We still didn't have a microorganism strain. We hadn't ordered it because we were dumb bachelor students. And so we went around Sherbrooke asking friends if they had kombucha scoby. You know, the tea kombucha. Because we knew that there was one bacteria in there that could actually produce this material. And so we ended up going all around town. We brought back 4 samples to Vincent's apartment, isolated them in his apartment in the kitchen. And the next morning we used one of those samples to start our culture. And to this day, 5 years later, we still use the same strain. That is the strain that is in our freezers in the lab, just because we've got some amazing results out of it.
Welcome to NGBiLab, a podcast about innovators and disruptors who are creating the next generation of companies in Canadian life sciences. Hi, I'm Jim Wilson, and my guest today is Jérémie Chaussé, who is the co-founder and CEO of AxCell Labs in Sherbrooke, Quebec. Jérémie and his team have created biomaterials that help regenerate body tissue after trauma from an accident or surgery. Made from bacterial cellulose, the biomaterial they've developed provides a new benchmark for subcutaneous tissue generation in surgical procedures, and it might just be the basis for the next generation of reconstructive surgery. What you're about to hear is that AxCell Labs did not begin as a traditional startup. In fact, the company was started by 2 university students who at one point found themselves going door to door looking for kombucha, but not for the reason you might guess. Before we get to our conversation with Jérémie, we'd like to mention this podcast is part of Next Great Big Ideas, Canada's Life Sciences Summit. NGBI is a national networking event for Canada's life sciences community, and we hope you can join us. For details, please go to nextgreatbigideas.com. This episode of NGBiLab was recorded in 2024. I am very pleased that our guest today is Jérémie Chaussé of AxCell Lab in Sherbrooke, Quebec. Jérémie, welcome to NGBiLab.
Thank you. Happy to be here.
I'm delighted to have you join us. So your company was founded in 2021, and I would appreciate you telling us what it's all about. Like, what is the problem you're trying to solve?
At AxCell Lab, we're focusing on soft tissue defect. So sometime a patient's going to present with a lack in soft tissue. It can be after a trauma. It could be after an operation, after a cancer, something like that. And so surgeons are often going to have to resort to implant fillers that are going to provide a scaffold for the body's own cell to colonize and replace this emptiness, right? Does that make sense?
If I can understand it, you're doing well. So
okay, awesome.
Keep going. So when there's an operation or an injury, you provide a substitute for human tissue until that tissue regrows.
Yeah, exactly. Yeah, exactly. And the word in English is way better than the one in French for those type of material. They're called scaffolds. Okay. So it's the same way as in construction, where they're going to build a scaffold around what they're building. Well, our material is a scaffold for the body to reconstruct, to recreate its tissues.
So you've got 3 people on your team at the moment, is it?
Correct. Yes.
Who else is on your team?
So my co-founder since the beginning has been Vincent Daniel Girard. We've done our bachelor in biotech engineering together here in Sherbrooke, and started this project during the bachelor, gone into our master degree together, also in biotech engineering, which we didn't in the company. That was quite funny. We kind of hired ourselves there. And just recently we had Jordan Mailloux join the team as a scientist in R&D. It was this August, and he's already doing great, and he's helping a lot.
Oh, fabulous. So you mentioned the idea hit you guys when you were doing your Bachelor of Science at the University of Sherbrooke. Could you walk me through that? Like, how did you come up with this?
We're kind of the wrong type of startup, because usually you're going to start with a problem and try to fix it. Me and Vincent were just doing our Bachelor in Biotech Engineering. We had this class. We wanted to work on this material that Vincent had heard of, that is bacterial cellulose. And so we asked the professor if we could work on this project, work on trying to produce this material during the class. He agreed, and so we ended up in the lab trying to produce it. And there's a funny story at how it all started. In biotech engineering, when we do a fermentation, right, microorganisms are producing our biological material, right?
Right.
Well, you need the microorganism to produce it, right? You can't just, ta-da, it's going to happen. So I think it was the evening before we would have this first day in the lab. We still didn't have a microorganism strain. We hadn't ordered it because we were dumb bachelor students. And so we went around Sherbrooke asking friends if they had kombucha scoby. You know, the tea?
Kombucha?
Kombucha.
Like the tea?
Yeah, exactly. Yeah, exactly. Because we knew that there was one bacteria in there that could actually produce this material. And so we ended up going all around town. We brought back 4 samples to Vincent's apartment. We isolated them in his apartment in the kitchen. And the next morning we used one of those samples to start our culture. And to this day, 5 years later, we still use the same strain. That is the strain that is in our freezers in the lab, just because we've got some amazing results out of it.
Wow.
Yeah.
Necessity breeds innovation.
Talk about luck, right?
That's fabulous.
And maybe to come back to your question, that was about how we actually got into this, is that after we produced it, we did the class on it, our teacher that was kind of mixed with entrepreneurship saw potential for us to meet some clinician here in Sherbrooke, and they kind of started pointing us in that direction.
So your team of 3 people, still based in Sherbrooke at the University of Sherbrooke, correct?
We are not based at the University of Sherbrooke
okay.
anymore.
Where are you now?
We are now in this wonderful office that we use half of and turn it into our own wet lab. We're still based in Sherbrooke, but just not in the university installation anymore.
Are you right downtown in the incubator there, or are you elsewhere?
No, we're not downtown, but we are in town. Yes.
Oh, great. Now, I had the pleasure of interviewing another group from the University of Sherbrooke who recommended we speak to you, Julie and the team at Tatum Bioscience, and they're doing fabulous work. And they invited me to Sherbrooke last summer to visit them. And I'd never been to Sherbrooke, and I was blown away by the campus and the city. It's fabulous. Forgive me, but I was surprised at how vibrant the life sciences community is there, and your evidence of it. Again.
Yeah, well, there's not much else to do here in Sherbrooke apart.
We just work.
We have good restaurant, we have good breweries, and that's about it. So when you come out of the university, either you start your own project, you start your own company that you're going to develop into something, or you're going to move out. And I don't want to move out.
Yeah, and it's what, about an hour drive south of Montreal?
Yeah, something like an hour and a half south of Montreal.
Okay, and it's beautiful rolling hills. I fell in love with the region, and I got to come back.
You're welcome back.
Well, thank you. So I'd like to talk a moment about your journey. Like, are you bootstrapped at the moment? How are you doing financially?
The initial strategy and the reason we did the master in the company was that we felt kind of protected doing that. We had access to university resource. We were kind of this little college cocoon at the start. So we probably had one of the longest master degree ever. I think we took 3 years to do our master, but we were leveraging all of those resources, all of the access we had to grow the company as much as possible into eventually a funding round, which we just did a first close. We're still looking for a bit more, but we now have some capital to keep going. And then after that, with the funding we received, we apply to everything. Like back in our master degree, we're doing every type of contest, every kind of pitch competition, everything just to get a little bit more and leverage this into our development to reach that step where we could actually go into a funding round.
When we come back, Jérémie talks about how AxCell is positioning itself in a competitive biotech landscape and why being a limber startup is a key business advantage. Stay with us. I'm Jim Wilson, and you're listening to NGBiLab, Canada's life sciences startup podcast. If you're a new listener, we would like you to know that we produce this and our other podcasts to promote Next Great Big Ideas, Canada's Life Sciences Summit. NGBI is an annual national networking event for innovators and disruptors, for-profit and not-for-profit leaders, members of government, startup founders, investors, industry suppliers, and academic entrepreneurs in the Canadian life sciences community. NGBI is truly a national networking event, and we invite you to join us. For more details, please go to nextgreatbigideas.com. Thanks for listening. Let's get back to the show. So the field that you're in,
Yeah.
I'm guessing, does not have a lot of other competitors.
You mean AxCell's field or biotech in general?
AxCell.
Well, it's a weird thing because you know what I was saying with we had a solution before having a problem.
Right.
So we still have this flexibility right now of choosing where we're going based on the stage of the company. We have a great understanding of the implant. We know it works to replace soft tissue. Now, what's left for us is to choose which one we're going to replace or where we're going to replace them. Soft tissues are everywhere in the human body, but depending on which one you replace, it's a whole kind of different application. And it does affect the competition.
So it can be inserted in the body cavity and sutured up?
That is what we've done with our animal model. Yes.
And are you near or are you in clinical trials yet?
That was the objective we gave ourselves with this round. So the round we just raised is to reach those clinical trials. So we want to finish all animal trials, all preclinical trial, and the goal for us is that in 2 years we are ready to start clinical trial.
So how much money do you need to get from where you are to where you want to get to?
Considering what we've just gotten,
Sure.
we were raising a little bit north of $800,000. We're combining this with a lot of non-dilutive funding.
Uh-huh.
And so right now we're not missing much. I'm still open to raising a little more just because we want to be safe. But right now the math is working. We almost have everything we need.
So if there are any listeners to this conversation who have got a wallet they would like to open up for startups that catch their ear, should they be contacting you?
Yeah, I'm happy to pitch to whoever's interested. We're still open to raising a minimum of 50,000 Canadian dollar. We're open to raising more, and it's not a minimum check size. So yeah, anyone who would be interested in learning more about AxCell, we're ready to pitch. We're ready to share documents, and we're ready to answer questions.
Okay, thank you for that. Now, I'm again assuming that your target client would be hospitals.
Yeah.
And perhaps the obvious market for you is the larger market, just a little bit south of Canada. Are you making inroads there yet, or is your focus provincial at the moment?
Our focus is provincial at the moment, at least for the preclinical stage of the company. So everything that is animal based is going to get done here in Quebec for now. Our current milestone right now, our current objective are based on advancing with the FDA. And after that, FDA versus Health Canada, they're quite similar. Health Canada doesn't like when we say if it goes through FDA, it's going to go through Health Canada. But we'll have enough data to justify our submission to Health Canada as well.
Okay. Now, the trials that you're doing currently, are they being done in Sherbrooke? Or
We're starting animal model early 2025, and our goal is to do preclinical trial 2026.
Okay. The fun part about doing this podcast is speaking to, in my mind, really cool people like you who are working on really cool ideas. And I'm wondering what your entrepreneurial journey has been like so far. Like, has it been what you expected, or has it been all over the map?
It's been all over the map. It's something I usually keep talking with Vincent, my co-founder, is how much we learn doing this. Right, our master degree was extra long. We took 3 years, but there aren't many engineering students that graduated from a bachelor or a master degree that have all of this knowledge. You know, the real challenge in industry, you have to understand fiscal stuff, right? You have to understand law. Everything is crossed in between, and having a decent understanding of how it all plays out together, I believe makes you an invaluable asset if ever AxCell Lab would exit, if we would go to do something else. We learned so much while doing this because we didn't have the choice. And as a small startup, you can't hire someone to do it for you. You don't have the capital for that.
Right.
So you have to learn it all by yourself. I must have been the the only graduate student that was working on his company taxes in the, in the graduate student office.
Well, that's probably a good problem to have, I'm guessing.
Yeah, it is.
Um, so has anybody kind of taken you under their wing and helped shepherd you along this journey?
Well, first of all, our professor from that first class at the bachelor was incredible help. We would have never gotten here without him.
And sorry, who was that?
Professor Patrick Burnett here in the University of Sherbrooke.
Well, shout out to him.
Yeah, definitely. He allowed us to do our master's degree with him. He was our research director. He allowed us to grow AxCell so much while still being master students, and always kept giving us advice pretty much for free, just as a general mentor. Then, of course, there was our incubator, the Asset here in Sherbrooke. I would say those two have been some of the big ones. Plus, of course, the clinician that helped us start the project, which are still with us today.
You one of the questions that I have is, who came up with the company name, and does it mean something specific?
Who exactly came up with it? I'm not sure. I remember sitting at the cafeteria at the university with Vincent and some other friends, and we were kind of like brainstorming a name, trying to play around cellulose, and we eventually came up with AxCell.
Yep.
The labs is pretty much just for the sounding of it, but AxCell has a lot of hidden things into it. The A and the X are for the microorganism we use. So the microorganism we use to produce cellulose is called Acetobacter xylinum, which is why the A and the X. And the reason why the X is lowercase is because in Latin, when you name species, the second word is going to be lowercase, the first one is going to be uppercase, and the cell is basically for cellulose.
Okay.
And we felt very stupid when we saw all the other companies that were working with this material that were also doing some little word plays with cellulose and X or the letter A, and that we're playing around that.
So was this the first iteration of the company name, or the last one?
It was the first and last iteration of the company's name. We haven't changed.
Oh, good. So
Yeah.
so the green A and the stylized X behind it.
Yeah, exactly.
With a stylized C.
We've touched the logo a little bit since then. Just we haven't changed the color, the name. We've played a little bit with the format or the style of the text, but that's pretty much
clever. I like it.
Yeah.
I asked earlier, has it gone as you expected? But perhaps the flip side to that question is, has there ever been a time where you thought, yeah, no, should we keep going? And if so, what pushed you further down the path?
It was during our master degree. If you ever read anything online in literature, like scientific student would do, on bacterial cellulose, which is the biomaterial we're using, I can bet you in the abstract it's going to say somewhere that it is biocompatible, that when it's implanted, everything's going to go well. And so we believe that. And Vincent and I objective during our master degree was to get in vivo data, which means going into animal to know what's going to be the behavior when implanted. And so I think we worked for about a year and a half getting everything approved. See, that's an example of when you have no clue what to do, but you got to learn it by yourself. So we had to learn how to do an animal protocol, how to get it approved, go through all the ethical committee, all that kind of stuff, which took us a long time. And when we eventually got there, did the test, we had a huge immune reaction. It really didn't go well. We didn't cancel the test, but we canceled the rest of the test just because we had to go back, and we went back to the drawing board trying to figure out what was the problem. And it took us about 6 months to figure it out and fix it, and that actually became our biggest, I would say, added value at AxCell, is that we fixed this immune reaction that we had. We went back into animals 6 to a few months later, and then we had the amazing result that we still had, and that pushed us into doing this funding round. But during the 6 month when we didn't know, that was very difficult.
Hey, it's Jim. We're going to take a short break, but when we come back, Jérémie shares what kept them moving forward after a major setback and what it takes to steer an early stage medtech company through uncertain waters. Stay tuned. I'm Jim Wilson, and you're listening to NGBiLab, Canada's life sciences startup podcast. I mentioned earlier that we produce this podcast and our other shows to help promote Next Great Big Ideas, Canada's Life Sciences Summit. But that's not the whole story. NGBI is also an independently organized, not for profit fundraising event in support of McMaster Children's Hospital in Hamilton, Ontario. Mac Kids is one of the top critical care pediatric hospitals in Canada that serves families and patients in need from Niagara Falls through Hamilton, Kitchener-Waterloo, and north to Owen Sound in Grey County. If you're interested in supporting a great cause, we hope you will consider supporting Mac Kids. For more information, please go to hamiltonhealthsciences.ca/McMaster-Children's-Hospital. Now you know our why. Let's get back to the show. There must have been points when you're in the lab late at night thinking, like, are we going to get through this?
Yeah, that's something like that.
So what drove you? It was just persistence and darn, we're going to figure this out. Or before we hang this up, I want to know why.
It's more that, and we've always told ourselves that we were two students that were founding this biomedical startup. You know, we would go into those entrepreneurial class, and when we'd say like, yeah, no, the sector we're going after is the medical industry. We're not stupid. We see those looks, right? The chances are of success are really slim. There still is so much risk, but going into the master, we always told ourselves that the worst thing that's going to happen is that we're going to end up with a diploma. So this was, I'm not going to say kept us going, but it was kind of like we're not doing all of this in vain. And what definitely did push us is that we wanted to know. We had to know.
And once you got over that hump, everything started to fall relatively into place.
When you beat a problem, there's always going to be a new one. And I had one engineering teacher that gave us a great illustration of what engineering was in general. It's you knocking your head against a brick wall. And if you knock it long enough, at one point you're going to get through or break the wall. So we knocked our head off.
And you got through.
So far.
Yeah, there you go. So let me turn that question around. What's been the biggest win so far?
I would say the moment we got those animal results, the second time was a huge win. And one thing that Vincent and I have always told ourselves is that if I go back to the story when we went around Sherbrooke to get those kombucha sample and
I love that.
incubated the next morning, we should have known that being so lucky at the start was a bad sign for what was coming.
How did you know to get the kombucha? Like, you just knew the ingredient? You thought, okay, it's going to be here?
No, we knew the type of microorganism we wanted was supposed to be in there, and we went off that assumption. And funny enough, a few years later during my master degree, I eventually fell on a new science article that was saying that isolated strain from kombucha scoby produces more cellulose, and I thought that was hilarious because we knew 3 years ago.
Well, things happen and don't for a
Yeah.
reason. So, what's the plan for AxCell Labs in the coming year?
So we're in this great position right now after closing a round where we kind of have the freedom to actually go and start doing more stuff without the capital constraint. So the two big things that are coming up are the animal study that we're talking about that's going to start in 2025. And the goal of this study is really to put our material into select clinical case and identify the most promising in terms of result. So basically, we have a lot of insight right now. We've done so many interviews with different clinicians. We have a list of application that we order, we prioritize, and we've developed our material for those application. Now we're going to go test it in actual clinical case. And the goal of this is to end up with only one, choosing one to push forward. Then we do the regulative aspect, which are the 2026 tests, also an animal. And then if everything goes well, in 2027, we're in human and starting clinical trials.
And how long, if that time frame is met, how long would the human trial be?
It varies. It's a minimum of a year for sure that we have planned right now. Of course, this doesn't account for the preparation and all the back and forth. Yeah, at least a year, and then we might do more than the one phase. But it's weird in med device to articulate the amount of clinical trial you need. The good thing though about those clinical trial and why we really want to get there, and that is our big objective, is that the moment we prove that our implant is safe in human is when we can kind of reopen the gate to many other application. The reason for that is, see, right now we have to choose only one to pursue. But the moment we prove it's safe in human, then we can start looking at other companies for them to develop it in different industry, right? In different medical device application. So
That,
we're
that's cool.
really looking for those human data.
What an exciting journey to be on. And you really are closer to the beginning of your journey, obviously, than from the end of it. Is there any advice in particular you wish someone had kind of pulled you aside and said, hey, you need to know this? And you mentioned that you had to learn everything, but was there anything in particular where you thought, geez, I wish somebody had told me this?
Tell everyone, as a technological startup, it is really important to clarify who owns the IP, especially in the case you're coming out of the university. Uh, it was more complicated than we thought it was going to be. We eventually managed to get it. Tackling this question early is the best way to go through this.
So you have patented the technology on which you're working.
Well, the first question was who owns it, right? When you're developing something inside of an academic institution, especially as a graduate student, there are some constraints around that, which makes sense. You're using university resources. It's a partnership in between you, your company, and this institution. But on the other side, if you're going to raise money, the first question that investors were asking me was, who owns the IP? Do you own the IP? What is the structure around it? And so we managed to eventually figure it out. We got it, and now we do have a provisional patent that's been filed. I think it was beginning of October. So we're on the right track for this. But I would have liked to know this earlier.
Is that kind of the same advice you would give others who might be considering following in a similar vein? If you're going to go down this path, obviously make sure that you can, from an IP perspective, that there's actually a problem that people want solved. Is there anything else?
Something that quite worked out for Weinstein and myself with AxCell was to really apply to everything we could in terms of contests, programs. We would apply to everything. And at some point, you're going to start to be able to reuse some of the material that you already created for those different contests. That really helped us, because that helped us gain initial capital that we then kind of leverage with non-dilutive funding during our master degree, and it allowed us to keep going.
And so the in vivo, hopefully leading to human trials and perhaps some really cool announcements by the end of 27, 28.
I think we're going to have them before the end of 2025.
Okay.
I
Okay.
hope so.
Well, I would love to have a follow-up discussion to hear how it's going and whether you pivot
Yeah.
and the reasons behind that. This is so cool. I love the idea.
Thanks.
And I really appreciate you joining us today.
Absolutely.
There's a lot of cool stuff going on in Quebec and in Sherbrooke. And thanks so much for your time.
My pleasure.
That was Jérémie Chaussé of AxCell Labs in Sherbrooke, Quebec. To learn more about the work Jérémie and his team are doing, check out axcell-labs.com. That's AxCell-labs.com. If you'd like to follow this podcast and our other shows on social, we are at NGBI Podcasts, and you can learn about the NGBI Summit at nextgreatbigideas.com. For information on who I am and what I do when I'm not hosting podcasts, go to jim-wilson.ca and laboccupier.com. You can also follow me on social at laboccupier. This episode of NGBI Lab was researched by Xenia Pinto and edited by No Limit Marketing. If you'd like to get in touch, send us an email. My address is jwilson@leonard, that's LENNARD.com. Thanks so much for listening. See you next week.